Good evening, everyone, and welcome to the third season of Scientifically Speaking, an interactive online lecture series that connects world-renowned scientists to high school students and teachers. Carefully curated to inspire, excite, and ignite young minds in the field of scientific research, this interdisciplinary lecture series aims to help students develop a creative way of looking at life and connect their learnings to address real-world problems. Before I introduce our speaker and moderator for the first lecture, I would like to give you a brief overview of Scientifically Speaking Season 2. Spread across 10 unique scientific topics ranging from planet Earth to green chemistry, artificial enzymes, motor proteins, and language processing, amongst others, Season 2 received over 85,000 registrations and saw the participation of more than 12,500 students and teachers from across the country. Season 3 of Scientifically Speaking promises to be more enriching and diverse. We hope to cover 6 interactive sessions where we bring to you some of the most interesting topics presented by renowned scientists from across the world. Our speaker for the first lecture today is Dr. Uma Ramakrishnan, molecular ecologist and professor at National Center for Biological Sciences, or NCBS, Bangalore. I would like to inform our audience that this season will be live across Zoom and YouTube. To moderate this session, we have with us Ms. Arnavaz Kola from the Cathedral and John Connon School, Mumbai. Arnavaz started her teaching career with D.Y. Patil International School. She then furthered her career as a chief genetics officer at Nutritional Genomics, where she developed an online course based on nutrigenomics for professionals in the health and fitness industry. She is currently teaching biology and environmental systems at the IB and IGCSE levels at the Cathedral and John Connon School, Mumbai. Arnavaz is a double graduate in human and molecular genetics from Virginia Commonwealth University, USA, and in biotechnology from Ram Narayan Ruia College, India. Welcome, Arnavaz, and thank you for taking out time to join us today. I request you to please take over from here and introduce our speaker for the day.
Thank you so much, Parneet. A very good evening to our audience. It is an honor to be here today to moderate Dr. Uma Ramakrishnan's lecture. Before we begin, I would like to thank Ashoka University for organizing this third lecture series on cutting-edge scientific topics. Dr. Ramakrishnan is an Indian molecular ecologist and a professor at the National Center for Biological Sciences, Bangalore. Her research investigates population genetics and evolutionary history of mammals in the Indian subcontinent, including work to save India's tigers. In 2019, she was elected as a fellow to the Indian National Science Academy. Dr. Ramakrishnan did a Bachelor of Science in Physics, Chemistry, and Mathematics, and a Master's in Biotechnology. She subsequently received her PhD from the University of California, San Diego, following which she was a postdoc at Stanford University. Dr. Ramakrishnan joined NCBS in 2005 as an assistant professor. Her laboratory developed methods to conduct population monitoring and population genetics with tiger fecal samples. Dr. Ramakrishnan has worked with the tiger expert and former director of Wildlife Conservation Society, K. Ullas Karanth. Together with Karanth, Dr. Ramakrishnan's lab used genetic sampling to estimate the tiger population in Bandipur National Park. Her research on the connectivity of tiger populations in central India was presented in court to stay the widening of NH7, which cuts across the Kanha-Pench corridor. Dr. Ramakrishnan, a very warm welcome to the first lecture of Scientifically Speaking Season 3. We're all enthusiastically waiting to hear from you. Over to you, Doctor.
Thanks so much, Arnavaz, and thanks all of you at Ashoka University for this invitation. And really happy to see so many students here. And I'm going to go ahead and share my screen. So the title of my talk is how will tigers survive the 21st century? And to a large extent, this is actually in all of your hands. And in a sense, all of us researchers will pass the baton of conservation on to you, the younger generation. So before we get started, it's always fun to remember, what tigers are. They are the largest carnivore that walks on Earth, which is alive, right? And a male tiger, for example, can be up to 4 meters long, while a female tigress can be about 2 to 3 meters long. A male can weigh up to 300 kilograms, and a female, which is smaller, can weigh up to 170 kilograms. So they are really large animals. And you can see this picture of a tiger in the background of Ranthambore National Park. This is basically a fort which used to be a hunting lodge, but today it's kind of in the domain of the tiger. Tigers rest here in the summer. So before we go on to think more about tigers, we should zoom out and as good scientists, really consider the status of biodiversity on our planet. So, biodiversity is critical for our well-being. And I think this is something which has hit us particularly hard in the last 1.5 years with the pandemic, reminding us about how an imbalance with nature can really devastate all of our lives. And, it's not just that tigers are beautiful and they are large and charismatic, but clearly the habitats that tigers occupy, the ecosystems that they live in, provide us with air, water, and, pollination, fruits that we eat. Without nature, none of these very simple services, climate regulation, water, and pollination are possible for us at all. And it's not just, that these habitats contain biodiversity. The bottom line is that biodiversity itself is under threat. Many of you would have heard of climate change, and we all worry about the changing climate and global change. But equally important, and this is repeatedly coming out in studies recently, is the loss of biodiversity. It's a big concern for our generation. And several studies have shown, for example, that 1 in 4 mammals is threatened with extinction. Extinction basically means when no individuals of the species remain on Earth. And why are species threatened with extinction? Well, for example, with a species like tigers, there has been in the past a lot of hunting, right? We have lost many tigers. The numbers of tigers have gone down because of hunting. And this is a picture from the National Geographic Collective during the British Raj times. Tigers were, I guess, much easier to hunt at that time. But also ongoing traffic of tiger parts. So this is a picture of, the tiger skins being used to make purses and so on. So here's a quiz sorry, I should have prompted that. But, one question which we may ask often is how many species of tigers are there in the world? Can I keep going or should I wait? Oh, yeah, it's gone. Okay. So, okay, we'll come to the right answer in a bit, but there's just one species of tiger globally. Okay. Oops, sorry. Yeah. So basically, these various factors like the hunting of tigers in the past, ongoing hunting today can result in fewer tigers, and that can push the species, for example, towards extinction. This is quite simple to understand. But also, tigers have not just lost numbers, but they have also lost their spaces. So imagine a big city with lots of people and lots of houses. People live in houses, and then you obliterate 95% of the houses, right? So there's no place for people to live anymore. So similarly, tigers have lost about 95% of their range. They're not like a very specialized species, which you might expect might go extinct. Tigers are very generalist. They live from, the Russian Far East, where it's really cold and it snows, all the way down to the tropical rainforests of Sumatra, Indonesia, and the dry deciduous forests like in central India, the Terai and so on. So they live in a variety of habitats, and they used to also live in this Caspian Central Asian area. So basically, a lot of this area has been lost for tigers, and actually subspecies, not species, but subspecies or populations of tigers have gone extinct since the 1930s, the 1940s, the 1980s, and most recently the South China tiger in the 1990s. So all of this together, the fact that the tiger numbers have gone down, the spaces that tigers live in. By the way, if you noticed, that's all in the last 100 to 200 years, right? These extinction of large areas where tigers used to live, extinction of individuals, local extinction called extirpation, like for example, the Caspian tiger, right? Or the South China tiger, all in the last 100 or 200 years. And so basically, because of this decline, we can classify species as endangered. And this particular species, the tiger, is classified as endangered by the Convention for Trade in Endangered Species and the IUCN, the International Union for Conservation of Nature, which is a body like the UN, which attempts to prioritize which species are close to extinction or they're doing okay and so on. So how do we study tigers scientifically? That sounds like so far we are just talking about things you read in the newspaper. But how do you study tigers to ensure that they don't go extinct? Well, from studies, theoretical work on extinction, ecological work, as well as field work on different kinds of organisms like butterflies. There have been many studies on extinction. What are the precursors, or what precedes extinction? When does a population go extinct, right? So basically what we understand from these studies is small populations are likely to go extinct. Isolated populations, populations which are not connected by movement to other populations, are likely to go extinct. You can think of those as islands. And populations with low diversity, with low genetic variation, are also likely to go extinct. So why the last one? Why does it matter if a population has low genetic variation? Well, there are two reasons why genetic variation matters. You can think of it as money in the bank for a rainy day, right? So sometimes when you have small populations with low genetic variation, you can have what's called drift or chance events, where by chance, because the population size is small, bad alleles are increasing in the population, and then the population is no longer fit or individuals do not do as well, and so they go, this population could go extinct. On the other hand, small populations can have inbreeding or mating between relatives, and that also tends to throw up higher frequencies of individuals who are having bad alleles, and as a result, those populations with inbreeding also have a higher chance of extinction. So this is our holy grail, that small isolated populations go extinct more often. They also tend to have lower variation and potentially inbreeding effects. So the question then is, are populations in India of tigers isolated? So let's just look at forest cover over the last 300 or so years in India. So this is India in 1700. The green is, forest cover or green cover, 1800, 1900, and 2000. So imagine if the tigers were associated, or correlated with this green. You could, of course, imagine that today, that's even 20 years later, they would be living on isolated populations, right? So in order to prevent extinction of tigers in India, sorry, this is especially important. Why? Because India actually has 2/3 of the world's tigers. Okay. So if tigers are to survive anywhere, they will survive in India, nowhere else. Okay. So if we are to prevent extinction of tigers globally, we have to protect them and prevent their extinction in India. So how do we go about doing this? What scientific information can be useful to prevent tiger extinction? Okay. Well, first of all, we need to know where the tigers are. We need to know how many there are. We need to ensure that populations are not isolated, that they are connected. And we need to make sure that they continue to be protected. They aren't killed because of some retaliation or some such thing. And I'll try to take you through small vignettes or stories of how our work has kind of contributed to some of these things. So how do we count tigers? Yeah, so maybe there's a good time for this question. What do you think? Do you think that tigers have unique stripes? Does each individual tiger look different from another individual tiger? So it turns out, that's good, that they are unique. It's pretty cool, right? Just like your fingerprint is unique, what makes tigers really unique are their striping patterns. And because their striping patterns are unique, if you study these striping patterns, you can identify or distinguish one individual from another. And there's a really fun picture of a cub looking right at a camera trap. You can see in the far background, this gray thing, that's a camera trap as well. So what you could do is in the forest, you can place these camera traps and then you can get pictures of tigers as they walk on this road. So basically what happens is there's an infrared beam between the camera traps. As the tiger walks by, it breaks this beam and the camera is triggered. And so there's a picture then of the tiger. And the person who pioneered this approach of using camera trap images and the information therein to estimate the number of tigers in a park is Ullas Karant. And here's a nice picture of him and some of his excerpts of his work from Scientific American, where he basically worked in Nagarhole National Park, Tiger Reserve, and other parts of the Western Ghats in Karnataka to actually take pictures like this of many tigers, and initially compared by eye and later helped work to design a software to distinguish individuals from each other. Once you know these different individuals, you can then use a probabilistic approach, a mathematical statistical approach to say, I saw this tiger once, I saw this tiger again, I saw this tiger only once, I saw this tiger 5 times, and use this capture recapture framework to estimate a potential number of tigers in a park. And this is an amazing kind of a innovation of putting together a method with a statistical approach to estimate the numbers of a very rare, generally a rare species. And after Ullas developed this approach, it was taken forward by the government of India and the National Tiger Conservation Authority. In fact, very recently, the last update on the numbers of tigers earned them a recognition from the Guinness Book of World Records for the largest conservation effort on the ground. And maybe we can have the question on tiger numbers now. So given that, basically India and the government of India has been estimating the number of tigers for many years now. And at the last estimate, can you guess how many tigers India actually has? Not bad. So actually there are about 3,500 or so tigers in India. These are approximate numbers, of course, they're estimates, right? Not exact numbers. But yeah, so basically there aren't that many tigers in India. So imagine if 60% or 70% of the world's tigers are here, there aren't that many tigers all over the world left. Okay, so basically, so far I've just been talking about, how we need to know how many tigers there are and whether tiger populations are connected, because understanding this will allow us to think about whether a population is prone to be going extinct. Now, one way to do this is with camera traps, but another way to do it is with DNA. And every one of our cells, as you all know, has a nucleus, and in the nucleus is jam-packed our genome. Why jam-packed? Because our genome is really, really long. If you open it out, it's really long, but it's like a big book, okay? And the tiger genome, for example, is 2,400 million letters long. So it's a book that is 2,400 million letters long, okay? And basically every tiger, just as it has unique stripes, has a unique genome. So if we could actually look at either the whole genome or specific words in the book, that is the genome, which are different between different individual tigers, we could potentially count tigers in a very different way using their genetic information. Okay, so basically what I do, and with, of course, my students and postdocs and colleagues in my lab, is we use genetic approaches. We use a genetic lens to study tigers, to understand where they are, how many there are, and whether populations are connected, and what can we actually do to protect them. So we call this field molecular scattology, because you can imagine going into the forest and finding a tiger and trying to get its DNA either from saliva or blood or something would be very, very challenging, right? I mean, I don't want to be in that situation where I'm wrestling a tiger. So what we do though is we just walk on these forest roads and look for tiger signs. In this case, it turns out, luckily for us, the tigers walk on these roads. They don't go through the bushes as much. They rest in the bushes, but they walk on these roads and they mark their territories, saying, this is my area, with fecal samples. And it turns out that these fecal samples can be a source of tiger DNA, right? Why are they a source of tiger DNA? Because when the feces passes through the intestine, it pulls off cells, epithelial cells. And then when it's deposited, you can actually see the epithelial cell layer. Well, you can't see the cells, but the shiny, mucus on the top of the scat, which what tiger poop is called, potentially has tiger epithelial cells and tiger DNA. Okay, so we can sample these. This is me several years ago in BRT in Karnataka, and we can sample these fecal samples, bring them back to the lab, extract DNA, and first of all identify, did this cat come from a tiger? Because we're not seeing the tiger, so we're not sure it's from a tiger. It could be from a leopard, it could be from a bear or a dhole, a wild dog. So we have to first check, is this particular DNA sample from a wild tiger, from a tiger or not? And after that, we can look at specific words in the genome to identify individuals and look at genetic variation. So just to give you again, a backdrop of India. This is India. This is the green cover, and in black are the tiger reserves. So you can see that they are quite isolated. And very interestingly, the median size or the median number of tigers in any park in India is about 40 to 50 animals. So by ecological theory, population sizes of 40 to 50 have high probabilities of extinction within the next 100 years. Okay, so unless these populations are connected, there is no chance of them surviving. And only 5 tiger populations, Corbett, Kaziranga, Bandipur, Nagarhole, for example, have more than 150 tigers. So most of the populations are very, very small in terms of numbers and also in terms of area. Only 4 to 5% of India is protected. The rest of it has people. It may have forest, but that forest is not protected. Okay, so as Arnavaz mentioned early on, this is soon after I started my job, we worked and collaborated with Ullas Karan, who was at that time doing camera trapping for tigers in Bandipur National Park. So what we did is we went along these roads, sampled scats, came back to the lab, identified individuals, and independently, completely independently, estimated the number of tigers based on these tiger genetic data. And it turns out that the number we estimated was almost identical to the number that Ullas and others at the same time estimated using camera traps. So that's good. Why? It's not that to say that there's any reason to doubt the genetic data, but showing from an independent way that you can validate what you find from the genetic data is very good. It shows us that these methods can work, that you can walk through a tiger reserve, pick up tiger poop, come back to the lab, not having seen a single tiger, you can estimate the number of tigers in a park. You can count them. Okay. On the other hand, what about connectivity or isolation? So I won't give you a lot of details, but we did work to try and understand whether populations of tigers are connected or disconnected. And what we found is that some parts or landscapes are already disconnected. So for example, the Western Ghats is already disconnected from the rest of India. And some parks are disconnected, like Ranthambore, and in other states, like in central India, for example, tiger reserves are well connected because of potentially movement of tiger individuals. Maybe we can have the last question here on Ranthambore. So basically, Ranthambore, I just said, some of you may have been there. It's a very famous tiger reserve. And it turns out that Ranthambore is actually isolated. It is not connected to other parks. So the question is, where is Ranthambore Tiger Reserve? It's actually one of the best places to. Good. So a lot of people got it right. It is in Rajasthan. It's close to Delhi, quite close to those of you who are at Ashoka. Okay. So basically, how do we study this? How do we study connectivity, right? It's okay, you can count the number of tigers, but how do you actually understand whether tigers are moving from one park to the other? Of course, the best way to do this would be to put radio collars on the tigers and monitor their movement. But how many tigers will you radio collar? And then, you have to radio collar some 100 individuals. And then you have to dart the animal, put it down, put on the collar falls off, all kinds of things, right? Logistically, quite challenging, right? So what we did though is something quite different. Oh, sorry, before we go there, what's interesting to think about is as we showed you the green cover, right? At the same time, you can actually think about India as distributions of people. So for example, if you look at India in 1992 and 2009, there's a huge change, and there's already so much older, right? There's a huge change in the number of people. So you can imagine, again, think about tigers. These are night lights, right? Think about tigers moving through, really literally, seas and seas of people. That's what they have to do. Can they do it? That's the question. So how do we study this? Well, first of all, we look for genetic signatures of mixing. Okay, so imagine you have two, populations, and population one has blue and purple genes, and population two has red and green genes. If these two populations are completely separate, they don't mix at all, you won't find any green and red genes here, right? On the other hand, if there's some mixing, you will find elements of the genetic makeup of population two in population one. It's that simple. So basically, if you go and sample all these populations in central India, not just one like we did in Bandipur, but sample all of them and look for how unique genetically are these populations. Are they mixed up or are they very discrete and separate? That should tell you about connectivity. And the second thing you can do is ask, if populations are connected, what are the habitats between them? So suppose two populations have, a lot of forest between them. Are they more mixed than, say, two populations which have a big city between them? Okay. So this took a lot of work, and real shout out to my PhD student, ex-PhD student Prachi, but she now lives in Delhi. She heads connectivity at the WWF India. Okay, so she and a team walked thousands and thousands of kilometers in central Indian forests. This is a beautiful picture from Kanha. And basically, the day starts early in the morning. Each team walks about 10 to 15 kilometers, and not eyes up, not eyes front, eyes on the ground, looking for tiger fecal samples. Because if you're going to walk that distance and miss it, that's a real waste. And in the eve, and as soon as you find a fecal sample, you collect it, you GPS locate it, and you have a data sheet. So this guy here, you can see he has a GPS in his hand. You have a data sheet where you fill in information. That's what here Kaushal is doing. All the information, sample number, and so on. Nowadays, we use an app for this, where the app automatically generates a lot of things and directly stores the information in a Google Sheet so that we don't, lose it, or it doesn't rain or some such thing. So when she collected all of this data and she came back to the lab and she analyzed it, what she found was really interesting. What she found was that overall, tigers don't have too much of a problem moving. The thing which makes it hard for them to move, they're just two things. One, high density human settlements, that's cities, not villages. And two, high traffic roads, not just any road, but high traffic roads, which means wide roads, 4-lane roads, 6-lane roads. Those are the ones with more traffic. How did she get this traffic data? Well, the Traffic Authority of India has data on traffic across all of these roads. So she was actually able to show it's a correlation. It's not a causation. That is, when there is more tiger movement, there's a lower chance of finding cities and high traffic roads. And what we did then is to project tiger connectivity 100 years into the future. And we showed that if there is unplanned development, that, we keep building more cities, building more roads with an unplanned way, where we put them doesn't matter. We just do it however we want. This increases extinction and lowers genetic diversity. Okay, so this was a very key result, which allowed for, as Arnavaz mentioned, the petitioning in court. So basically, this additional work there showed that extinction can really change based on whether you have connectivity or not. Okay, and as I said, as Arnavaz said, this is basically the Kanha on this side, and this is Pain Shear. And in 2016, NH7 was going to be expanded, all set to be expanded to a 6-lane highway. Okay, this is 2016. Our paper and results in it were then used in court, not by us, but by several people in NGOs, Anish Andheria, the Wildlife Conservation Trust, Milind Parivakam and Aditya Joshi, who are in Nagpur, based in Nagpur, but at the Wildlife Conservation Trust, to petition that NHAI, the National Highway Authority of India, must implement some mitigation. If they're going to just expand this road, that will lead to higher extinction of tigers in the next 100 years. And our data showed how much extinction would increase, right? So the simulations and the data was used to say, this is how much it will increase. And because that seemed very high, the court ordered that NHAI must implement mitigation, and the mitigation measure they implemented is this underpass. So wildlife can walk under, and the highway basically goes above. Now, you would think, why not just do this all the time? The reason is, when projects are planned, that is when these interventions have to be suggested. Once the project is planned, and then afterwards, if you start this legal process and suggestions, it's too late, right? In this case, we won that battle, but overall, there should be a better integration of science. We should have evidence and these type of data going in, not to say they should not be development, but to plan it such that it minimizes extinction of biodiversity. Okay, so just very quickly, I'll tell you guys about this very recent project we're working on. very exciting. So you might have read a lot about tigers coming out of parks, sometimes often eating cattle, wild livestock depredation, right? But also sometimes attacking people. And some of you may have seen this recent movie called Sherni, which I really loved. Thought it was fantastic, very realistic, very nice movie. You should see it if you haven't. So the thing is that this is a very beautiful camera trap image of a tiger eating a cattle, okay, outside the park. This is outside Bandhavgarh Tiger Reserve. And I'm sure you'll notice that the tiger is actually licking the cattle. If you have a cat, you'll know that cats also lick themselves, and they clean, similarly, they clean the prey before they eat it. So what happens when we have conflict, when we have tigers coming out and taking cattle or sometimes attacking people, we don't often know who is the tiger, which individual is involved in conflict. So what we are trying to do is to understand, using the like stuff left by the tiger, the genetic material, who is actually causing the conflict. So here's another picture, and my student Himanshu, he's a completely passionate, very devoted guy, completely crazy. And he actually even set up these sticky pads so that when the tiger comes to eat the cattle, there's a double-sided sticky tape here, and the sticky tape pulls off a few of the tiger's hair. So apart from the lick marks, which you will get on the cattle, you'll also get some tiger hair. You can imagine how much fun this field work is, going after dead, stinking cattle. As soon as a cattle dies, we usually get a call. He's distributed his number to all the villages around and rushes over and looks for all kinds of things on the cattle. So this is a jugular bite mark. So when the tiger kills, it usually attacks at the jugular, and you will be amazed to know that if you take a Johnson's earbud and swivel it around over there, we are able to get enough DNA to individually identify which tiger killed this cattle. So that's pretty cool. That's like detective work, right? So basically, imagine that each kill site is like a forensic investigation site. And at each kill site, you have lick marks, you have bite marks, you have fecal samples where the predator has eaten and defecated, you have pug marks, and a lot of times you have a lot of tiger hair, because as the animal is eating, it pulls off. A lot of hair is also pulled off. So basically what we're attempting to do is really identify tigers involved in conflict. Are there problem individuals? Are there problem places? What are the patterns and what are the trends? Because usually managers are just faced with one conflict situation and they don't know what to do about it, right? So for example, we have now collected 400 cattle kills, data from 400 cattle kills across 1 year in Bandhavgarh. And hopefully such studies will give us some insight. So I'll just end with a very fun example of international wildlife trade. I started with international trade saying that tigers are still under threat because of the hunting for tiger parts. So it turns out that this little cub is a 3-year-old or so, 2 to 3 month year old tiger cub was confiscated by the US Fish and Wildlife Service when a teenager was driving across the US-Mexico border with this tiger cub in a passenger seat. Okay, so they found this cub and they didn't know what to do with it. Okay, it's illegal to transport tigers like this, obviously. You can't take a tiger across borders without a permit. And so the first question was, who is this tiger, right? So basically the tiger was placed in the San Diego Zoo because it was unwell, the cub, but they reached out to us and said, you guys have a lot of genetic information on tigers. Can you take some DNA from this tiger and identify, is it an Indian tiger? Is it a Russian Far East tiger? Where is this tiger from? Okay, so basically remember, this was the distribution of tigers. And what we did was we were able to sequence at low coverage the genome of this individual. And when we threw it in with data from known Indian tigers, Russian tigers, and so on, for example, the figure you see on the right is what's called an assignment, like a genetic identity. So for example, Russian tigers are green. They have alleles which make them green. Okay, in a way, they have green, a set of alleles which look together a unique color, right? Bengal tigers, which we have a lot of data from, are blue in this plot. Okay, and tigers in the Malayan Peninsula over here, Southeast Asia, are red, and Sumatran tigers are orange. And the cub, for example, it turned out that this cub was a nice mix. It was 40% Bengal, 40% Amur, 15% Sumatran, and 5% Malayan. So this was not a very useful answer for them, because what happened was, because this cub was a full mixture, according to the American Zoo Association laws, they could not protect it. So they breed, the American Zoo Association only breeds Malayans, Sumatrans, and Amur tigers. Okay, they don't breed Bengals. And since this was a mixture, it couldn't enter into that pedigree, and it had to be put up for adoption. So this is an example where, like, a simple genetic, what seems very scientific, to do a genetic test to understand ancestry or identity, can actually have implications for the life of this individual. I should tell you that this tiger is doing well. It was adopted by a rescue facility. There are several of them. Like we have Goshalas, there are rescue facilities for tigers in India, in the US, and basically it's doing fine. So I just wanted to end with flagging, I think, a really big and very interesting question of our time. The last year, we have been enveloped by the coronavirus pandemic, which is a zoonotic disease which has potentially spilled over from bats. And this is a picture of a bat cave in the Western Ghats. And you can imagine why there may be high transmission and spillover of pathogens from such spaces. All those bright lights are bat eyes, and bats are packed in high numbers together. So you can imagine that, since interfaces between humans and wildlife are increasing, the potential for zoonotic spillover will also increase. And there's a lot of work which remains to be done to understand, how human exposure and risk will vary in the context of what's going on in biodiversity. And this is something which I'm hoping to work together with both Balaji and Kritika, who are faculty at Ashoka University. So I thought I would just mention, much of this work is almost impossible without collaborations and interdisciplinary teams. And also to mention, since you're all young people, that not to end on a very sober note, but the biodiversity and climate crisis is something which we all need to think a lot about. And we are hoping that work we have done with several others as part of the Biodiversity Collaborative has proposed a national mission on biodiversity and human well-being. And we hope that through the various aspects of this national mission, there will be scope for bringing back the balance between people and nature. So I guess I just should thank all of the fantastic people who have done all of this work, and the funding agencies who have funded my work. And I just stop there, and I'm happy to take questions. I hope I didn't run over time.
Thank you so much, Dr. Ramakrishnan. So while it is disheartening to, see how the tiger habitat and populations have declined over the years, but at the same time, one feels hopeful seeing the work your team is doing to ensure that tigers survive the 21st century. It's also fascinating to see how much information can be revealed from fecal samples, right? And your team uses a combination of molecular, computational, and statistical techniques to sort of retrieve and analyze that data from these samples. So before we, there are several questions lined up, several interesting questions. So I think we'll get started with those. The first one is, will molecular genetic tools be used for tiger conservation in the future? If so, what would the future look like?
Yeah, so Ananya, I think I understand the tone of your question. So for example, will we introduce captive bred tigers into the wild? Will we select for specific kinds of tigers with specific kinds of traits or mutations? And I hope not, but I have to say that I think that I'm pretty sure that the future will require a lot of management, in the sense that tigers are not going to survive on their own. They would have to be assisted, and this assistance may require selecting fitter individuals. A very nice example is, the Tasmanian devil. It's like this really endangered marsupial, carnivorous marsupial, and they have this contagious mouth cancer, which is devastating their populations. Now, recently, scientists found that some devils have a allele which protects them, a genetic variant which protects them from this cancer. So what they're trying to do is, in the natural population, to increase the frequency of this resistant allele. Now, that is also genetic management, right? So I'm just giving it an example, as those things, such kind of things may be what happens in the future. If we cannot maintain connectivity, we may have to airlift tigers to ensure that there is gene flow and a genetic variation is maintained, for example.
Okay. That's actually interesting about the Tasmanian devil. Okay, the next question is there a time limit between when the tiger scat is ingested and till when the sample is viable for genetic analysis? Does it decay with time, and how fresh should it be?
Yeah, definitely decays with time. I think as people have shown, even with ancient DNA, with things like mammoths and things like that, the most important thing is also environmental conditions. So if the environment is very dry and hot, it's okay. Best is cold and dry, okay, like permafrost. So that's why we are able to get DNA from Neanderthals and woolly mammoths and stuff, because they are preserved in a good way. In places like Europe, right? DNA is preserved well. Overall, DNA is much more stable than RNA. So overall, we are better off working with DNA in that sense, right? But in environments where the humidity is very high, it decays much faster. So, for example, our success rate from areas in the wet Western Ghats or in the Northeast is generally much lower, because the DNA, however fresh it is, it degrades much faster. The sample degrades much faster. In drier areas like Central India and all, what we look for as a practical tool is dung beetle activity. If we can see activity of beetles and stuff, then we know that most probably the sample will not work. Whether you collect it or not depends on where you are. If you're in Ranthambore, where the density of tigers is high, you will skip it. But if you're in a place where there's very few tigers, you'll take whatever you get.
So we're really dependent on the environment,
yes,
on whether we get good fecal samples. Okay, another question, I think this is with regards to one of the slides that you shared. In one of the images, the scat of two different tigers was seen quite close to each other. Does that imply that territories are overlapping? Or does that imply that they were just probably taking a walk and they just...
Yeah, that's just one scat probably. But basically, what you do have to be careful of is mothers and cubs. So sometimes when, mothers and cubs are always together, the cubs stay with the mother for about 2 years, 1.5 years or so. And at that time, you may see them defecating together, but they don't have latrines. Like, for example, other carnivores like dhole, they all defecate, they're group living, they defecate together. Okay, but tigers are not like that. They are solitary, they're not social like that. And what we do anyway to be cautious is, if we have one bolus or one piece of scat, we won't mix from another sample into that. We'll sample that and then we'll sample the other one, and then we always check it in the lab to see whether it's a different or the same individual.
Okay, lovely. Very interesting. Another question is, do albino tigers still exist? If so, are they not a different species? Can albino tigers return if they're extinct? I've heard that tigers become albino due to natural genetic modification. Is this true? So multiple questions in one.
Yeah. So, it's a great, Ryan, to see your questions. So first of all, I would clarify that there are no albino tigers. Albino means complete lack of pigment, which we do not see in tigers. We have a phenotype called the white tiger, which is the absence of orange pigment. Okay. The white tigers have stripes and they are white. Okay. There are several other color morphs. There's also a golden tiger, which we are trying to study in Assam right now. There have been some pictures of it which have shown up. And there is, of course, a snow white tiger, which is the closest to an albino, which is a double mutant of white and golden. Okay. So basically, so golden is the absence of, not the absence of black, but black becomes light brown. So a tiger is white, orange, and black. Okay. So in one mutation, there's no orange. In another mutation, there's very little black. Okay. And there's actually a third thing, which we have a paper coming out soon about, called pseudomelanistic tigers, which are quite black. And maybe you'll see this soon. But basically, all of these are mutations. That is, remember we said that there's 2,400 million letters in the tiger genome. Some of these letters or these words code for genes which affect how the tiger looks, affects the phenotype, right? And the spelling of that word has to be exactly precise. Now, in case there's a spelling mistake in that word, that gene and the protein associated with it is not okay. And as a result, there's a phenotypic change. Okay. So both the white tiger and the golden tiger, there are all because of single point mutations. One mutation, one spelling difference in the 2,400 million letters is enough. Now you said, are they different species? Not as far as we know. Why? The definition for a species, biologically speaking, is individuals of two species don't tend to mate with each other, or if they do, they have infertile offspring. Now that is a difficult thing to assess, but overall, if you just look at genetically how different our species, on an average, they are 5% or more different. Okay, for specific markers, but 5% is a lot. So 5% of 2,400 million is a lot of differences you would need, consistent between A and B, right? And that is not the case for white tigers. White tigers were originally found in the wild. In Rewa, there was an individual called Mohan, which was brought into captivity, and all white tigers in the world today are from Mohan. Now, continuing to have white tigers requires inbreeding, because there was very few white tigers to start with. And what you're doing is trying to keep on getting that spelling mistake in all the children. Okay. So you need to breed back. That's the only way to do it. And so zoos actually across the world have stopped breeding white tigers now, because inbreeding leads to other detrimental effects and the tigers are not healthy. So, sorry, it was a very long story, but as of now, we don't know of any white tigers in the wild, but other color morphs like golden and the pseudomelanistic tiger do occur in the wild. Mutations occur naturally. That's the point.
So, doctor, based on the information you shared, I actually have a question. Does the white tiger, the mutation that codes for the white color, does it have any advantage in the environment because of which we're breeding it, we're doing interbreeding, or is it just to sort of preserve that coat color?
Well, in the zoos, they're doing it because of the look.
Okay.
And then there's a lot of, and it's kind of like there's a lot of fascination for white tigers, I guess, or something like that. But basically in the wild, to answer that question, we would have to look at a study which is cross-generational, that we would have to see is the fitness or the reproductive success or the number of offspring of white tigers more than a wild type or a normal tiger in the same location, right? So these kind of studies would take time, and I'm not sure anybody has ever done anything very effectively to look at that. The thing to remember though is that tigers don't have predators.
Yeah.
There's no one who preys on a tiger. So unless the white tiger really stands out while it is hunting and then the prey is able to see it much better or something, maybe it doesn't have much enough of an effect. By the way, there is also some pictures which have surfaced of some white tigers in the Nilgiris from 2019, taken by some tourists. Whether they're real or not, we don't know.
Okay, lovely. That was great. The next question is there any method that tells us about the age of the tiger? Can one estimate the age of the tiger by looking at the stripes pattern?
So the age of the tiger, you cannot assess by the stripe pattern because stripes are laid during development and it's kind of a permanent thing. Okay. And unlike us, tigers don't white. I mean, their hair doesn't grow white or something. So they look the same when they're young or old. Okay. But there are some new methods where people are using telomerase length, some molecular marker, to try and estimate age. And they've done it in a lot of species. But for that, you need high quality DNA, and fecal material usually has degraded and low amounts of DNA. And so we did try a little bit once, not very hard, and we're not able to do it, not get it to work, but this would be a good approach to try to look at age.
Lovely. How reliable are these tiger estimates? So I think we did a poll regarding one tiger estimate, and how often are they carried out? Are there multiple techniques used to determine the tiger populations?
Yeah. So basically the thing is that you camera trap a certain area. Okay. Yes. And then you estimate the numbers in that area, and then you extrapolate to the rest of the area. Suppose you camera trapped in, 10% of the park. You're going to estimate for the rest of the park. So obviously, if the density of tigers varies a lot in that extrapolated area, then your extrapolation might be wrong, right? At the same time, you don't count certain areas. You don't have enough, you don't put in effort everywhere. So you're not able to count in certain areas. And so you're again going to make some extrapolation. So also the thing which a lot of people ask about is the increase in numbers. So definitely the increase of numbers, like 5 years more, so increase so much. That is something we should view with caution, because every time you're looking, your field effort is different. That is, before you were not looking in Odisha, now you're looking in Odisha. So like that. So if you're adding up more areas, you are going to get more tigers potentially. But all that being said, it is true that tigers have recovered post 1972 and Project Tiger, when Indira Gandhi took a specific interest in tigers. She was specifically very passionate about wildlife and took a specific interest in the Wildlife Protection Act and in tigers. Basically, the number of tigers has gone up. It's not that it's not gone up. Whether it's exactly 3,500, I mean, does it matter? I don't know how much it matters. It's more important to understand the directionality of the trend. And we should put in more robust approaches to assess trend. Yeah. We are not doing that right now. We're just saying how many. And we're fixated on the numbers, just like we're fixated on the number of positives and the number of this strain or that strain with COVID, right? Yeah. So we should understand the patterns and the trend also.
Great. Is there DNA of any other organisms found in the tiger scat?
Of course. So I didn't go into it, but for example, I just talked about the epithelial layer or the top part of the scat, but if you just take your forceps and pull out from inside, a lot of the DNA you get will be of the prey, but the prey DNA and the tiger DNA is minuscule compared to all the bacterial, fungal, and other DNA in the scat. So if you just take a scat, extract DNA, and then you sequence whatever is there, and you throw it at the universe of DNA sequences, it's called GenBank, less than 0.1% of the DNA is tiger. So most of the DNA in tiger scat is other stuff.
Do they also have like a microbiome like us humans?
Yeah, they do. I mean, we haven't spent too much time investigating it, but it's something which we were thinking about looking at. You know, so the thing about microbiome is that it's a very broad and descriptive thing. We can't really get a very, precise information about the different bacteria, but it'll be interesting to see whether, say, there are signatures in the microbiome of, I don't know, like isolation and different kinds of diet. Like we've just done a study where we looked at diet in Ranthambore, and it turns out that the most prevalent species in diet are cattle. This is supposed to be a wild animal.
Yeah.
So diets are also changing, and so too will microbiome.
Okay. This is again another question with regards to the scat. Does the texture of the scat give an idea about the health of the tiger? This is very interesting.
So basically, this is a lot of gory details of digestion and indigestion. But tigers eat once in 4 or 5 days because they eat big prey, like bison and sambar and all that. So as soon as they eat, the first scat is liquid. Okay. And thereafter it becomes solid, and all the bones and all come out. The hooves, the bones, and all that stuff. So they can't digest that. They just throw it out. But I have never seen a loose stool, shall I say, in all the scats that I have seen. But definitely we do see these first stools which come out post eating. I don't know though. What we're trying to do is, again, very exploratory and could completely flop, is there are known chemical compounds associated with certain diseases, right? Could be something like cancer. There are chemical ways to assess cancer, right? Chemical cues. And so we're trying to see whether we can get volatiles from, volatile chemical compounds from scat, and look at whether certain individuals have something, but it's very exploratory. We may not find anything.
Lovely. Can a tiger or any animal be cloned if we have their DNA with appropriate machinery?
Yeah. Sure. It's happening now. And that's probably one of the other, questions when we talk about management and genetics in the future, something to think about. But, the thing is that at some point we need to look at costs, right? So if you're talking about cloning, it's really expensive. And recently there's this huge furor about cloning the Przewalski's horse. There's a company called Revive and Restore, and in the US, and their mission is to clone species. And the Przewalski's horse was cloned. It was a wild horse. There are no wild horses left in the world. The Przewalski's horse was brought into captivity. It was recently cloned and mothered by a horse, a regular horse. And the Przewalski's horse is running around this new clone. And that's fine, and that's fabulous. But remember that at some point, these animals have to go back into captivity. And the famous examples of reintroduction kind of remind us that important also, or critically important, is removing the threats the species faced in the first place. For example, the Arabian oryx, bred in captivity, extensively managed, reintroduced in the wild, to disappear in a few years, again from hunting.
Correct.
So what was the point of that? So when we think about projects like cloning, we need to think it all the way through. By the way, our own project, we didn't clone the cheetah, but the cheetah is coming soon to MP, and because it was in India and is now extinct here. So there is definitely a lot of rewilding in the works and going on. But I think cloning is definitely a possibility, but it must be integrated with ecological knowledge and plans for reintroduction, which are ecologically sensitized.
I guess also with respect to cloning, when we talk about cloning, maybe the ethical constraints of it should be taken into consideration, because you start with one species, then where do you stop? Like, where do you draw the line?
Yeah, okay. So that's why this is a company. Revive and Restore is a company which uses philanthropic donations, and the kinds of regulations for companies are different from, for example, because they are stockholders which control these considerations, versus like if you're a university scientist or a research scientist, the ethical considerations or guidelines are different.
That's actually food for thought to think about cloning. That's like the next step. Okay, another question we have. How do you define an isolated population? Can you comment on it in terms of area and length? So I'm guessing the size of an isolated.
Yeah. That's a very difficult question. In fact, very good question, because this paper which I was telling you about, about the pseudo-melanistic tigers, the referees asked us this question because we kept calling it an isolated population. It's difficult to answer that question in terms of size, but I think that populations which are not, genetically part of a larger set of populations, right? And there doesn't seem to be landscape connectivity. There doesn't seem to be a good way for individuals to get there or out of there. We would, suggest that they're isolated populations. But we would have to look for signatures of isolation. Once a population is isolated, it would drift, or the genetic variation can go up and down, and it would lose genetic variation over time, and it would be subject to inbreeding and potentially inbreeding depression. So we can hypothesize that a population is isolated, and then we can look for signatures of isolation. The way to prove it would be to really show that it is completely closed. No individual has gone in, no individual has basically gone in. If individuals go out, it's fine. If there's no input, then it's still isolated.
So would that analysis sort of be over generations of the tiger population?
Yeah, to prove anything like that, it would probably take a lot of time. So, for example, I think, people are still surprised that Ranthambore is isolated, because it seems to be, and it's doing well. The tigers are doing well there so far. But, the Chambal, basically on the north, you have Alwar, the big city. And Sariska is right across Alwar, but it's not connected to Sariska, which is the closest tiger reserve. And the Chambal is to the, east of it. The west is, of course, desert. And the Chambal is a barrier, right, potentially to MP. The next population in MP is quite far away.
Okay, quickly moving on to the next question. What are the laws that protect tigers? How often are highways and settlements diverted to protect the tiger population?
Yeah, so basically, the laws that protect tiger, the Wildlife Protection Act, and we say even in the Constitution, as Indians, we will protect wildlife, and nature. And basically, the Wildlife Protection Act lists tigers as Schedule 1, which is subject to the highest protection. So any work that even I do or my students do, we have to take permission to enter the forest, to come out of the forest. What are we going to do in the forest and so long? And we have a lot of regulations, like we can't wander about at night, we can't do this, we can't do that. We can't collect blood samples of tigers very easily. It's very, very complicated. We have to go all the way to the center for permissions and so on and so forth. So actually, even doing research on species like tigers, because of the laws that govern it, is quite tough. But what was the second part of the question again?
How often are highways? I think this is with regards to your case for the N87.
Yeah. So basically, the thing is that there's an organization called the National Board for Wildlife. Okay, the National Board for Wildlife is chaired by the PM, and it's supposed to meet every time there's a project which requests for diversion of forest land or land which is encroaching into protected area. Okay, but the highway is outside, no?
Yeah.
It's in what is called potentially a corridor. The corridor is not protected. Okay, so then that has to be a different kind of battle. And so that's why there's a huge movement. A lot of conservation scientists are trying to say we need to talk about landscape protection, not about this protected area, that protected area, but the whole landscape. Of course, there's different levels of laws. Like obviously within a protected area, you can't poach animals, you can't pluck flowers, you can't do a thing, you can't kill an ant, nothing, right? Outside the protected area, you can probably, kill ants or whatever, So there have to be different levels of laws. but there should be some way to protect even those corridors, because tomorrow, suppose someone wants to build a big resort there, then what happens?
Right. So does it, I'm assuming it's based on the different zones of the national park, like the core
Buffer,
and buffer.
yeah. But what we are trying to do, so what we try to do in that simulation study is we showed that, how could we prioritize these corridors, right? And the kind of work we're doing right now is we're actually working, we've already done work with jungle cats, there's a small cats, wild cats, leopards, with dhole. We're doing work with sambar, other species, to say, do they use the same corridors?
Yeah.
Because you start by saying, oh, tiger corridor, you protect it. Then you would find out that jungle cats are using some other space. So whatever you protect for the tiger doesn't work for others, right? So we need to think a little bit ahead and say, let's use a multi-species approach. Let's think about all these species. But remember, when you're going to have corridors, when you're going to have tigers moving through human landscapes, you will have conflict again, right? So it's a complex problem. We want to promote connectivity, but promoting connectivity brings large carnivores and even species like elephants into very close, could bring into very close proximity. That's happening anyway. And so we need to really grapple with this new field, which is called coexistence. So how can we humans live alongside these animals?
So, Dr. Ramakrishnan, we have time for one last question. So, I'd like to ask you, what inspired you to get into saving the tigers and using your biological expertise? And can you just tell us something more about your work with them, please?
So, I guess, I'm actually, I'm inspired by curiosity. I'm just very curious about things. And I'm also I like things which are challenging. So if something is, like a lot of, as a scientist, you're supposed to investigate things which are the next frontier of understanding something in the field of whatever, right? But I just find things curious, and I don't care whether anybody else in the world cares about them, or if the other people, experts, think it's important or interesting or not. If it's something which is relevant and I'm curious about, then I would like to investigate it. And I think that these are very difficult things. Logistically, a lot of these things are very difficult. And so a lot of scientists, they don't want to investigate them because you might lose a lot of time in the logistics and all. But for me, I just love being able to work in different parts of India. That really inspires me, because as I learn about tigers in Odisha, I learn about Odisha, through that lens. So I'm really lucky, and I'm very lucky to also be at the Tata Institute of Fundamental Research, where this kind of science is, encouraged, and we are allowed, in a sense, to really, seek out whatever we're interested in.
Also, lastly, if a layman, like if non-science students want to get involved with wildlife or tiger conservation, how can they do it?
Yeah, so that's a question a lot of people ask. And I guess, the best way to do it is to, in some sense, be discriminating consumers. So, as citizens,